also ended up with discussing the vegetative reproduction, which normally happens either by way of fission, the budding, the fragmentation, and through which the asexual spores are produced. And we had a discussion about the major asexual spores which are normally produced by the plant pathogenic fungi, and also the type of flagella that these asexual spores do exist. Today we'll be talking about some part on the asexual fruiting bodies. The asexual fruiting bodies, as I already mentioned in the yesterday's class, the fruiting body, that is a fruit bearing structure or the seed bearing structure, can be called as a fruiting body. And often most of the fungi do produce the fruiting bodies. If you want to imagine how does the fruiting body looks like, it is a simple way to understand is you can imagine a mushroom. Mushroom is also a fruiting body. Okay, so but different type of fungi produce different type of fruiting bodies. So of which some of the fruiting bodies produced are, one of them is called as a cynema, is one of acervoli or acervulus, and a pycnadium, and finally the sorus. These are the major 5 types of fruiting bodies that are commonly produced by the plant pathogenic fungi. And how do they differ? They differ with respect to the morphology, basically. So how does this morphology varies? If you talk about the cynema or cynemata, it is also called cynemata. It is often also called as a cynemata. So this is a kind of a compact or a fused upright conidiophores. This is a conidiophore, upright conidiophore, and the spores are forming like a cluster like, or a cauliflower head, you can imagine, like a cauliflower head. And all these conidiophores are cemented together. These are cemented together, which are upright conidiophores. Okay, conidiophores, and the spores are forming like a head-like cluster or a cauliflower-like cluster. And often I said these conidiophores are cemented together. And the example for this type of asexual fruiting body, that is the cynema, is the ophiostoma. The example is the ophiostoma. And the second type that I mentioned is the Sporadicium. Is another type of fruiting body, which is often as a superficial growth on the surface or a cushion-shaped asexual fruiting body, where you will see the spores emerging out as a seeds. That is, they are superficial, cushion-shaped asexual spore and the conidia. The conidiophore and the conidia. So this will be in the clusters. This is similar to that of a cushion-like structure. A cushion-like structure on which you will see the conidiophore and the conidias coming out from this cushion-like structure. And this type of fruiting body is seen in case of Fusarium and Epicoccum. So another two important genus where they do produce the Sporadicium as a fruiting body. Okay. The third type which I mentioned is a Acerolus or Aceroli. Acerolus or Aceroli. This is basically a kind of errampant type and you can see that the conidiophores and the conidias emerge out. And often we see that these are often as like a saucer shape. This fruiting body is a saucer shape which is depressed, and it is basically the aggregation of so-called as the pseudoparenchyma, which is an aggregation which is resembling as similar to that of your parenchyma, which is a parenchymatous tissue, but a false parenchymatous tissue. And this is an errampant, means they emerge out from the epidermis and form like a saucer-shaped structure. And in some cases, they do also has a sterile structure from it, which is called as a setae, which is called as a setae. This type of Aceroli. The example for this type is the Colototrichum. The genus Colototrichum, as well as the Pestalo. Cushion-like fruiting body, having the conidiophores and the conidia, which normally break out through the epidermis, and also they can be seen as a sterile structure, like a setae is commonly seen. And this aggregation is because of a epidermis. Okay? And the fourth type which I mentioned is the pycnidium. The pycnidium. Pycnidium is basically a kind of a globose or a flask-shaped structure. Okay, a flask-shaped structure within the tissue. Normally, it is seen in the tissue of the plant. The tissue of the plant. That is, they are immersed into the tissue of the plant, producing the conidias inside. The conidias with the conidiophore. And the spores which are produced in this type of pycnidium, these are called as a pycnidiospores. These are called as a pycnidiospores, which are a flask-shaped or a globus-shaped fruiting body. And the last type of fruiting body is called as a sorus. Sorus is often produced in case of the rust and smut fungi. The rust and smut fungi, which is basically nothing but the compact of the sorus or the spores, compact fruiting body, where the spore masses of rust and smut, where they are filled in the cavities. If you consider this as the plant cells, so in the cavities, these compact sorus or the rust pustules, they are forming like a rust, like a compact fruiting body, filling the cavities of the infected areas. Okay, this is another type of the asexual fruiting body which is produced. Okay, and when we look into the fungal tissues, there are so many modifications that can be seen. That is the modifications of hyphae and the tissues of mainly the fungal tissues. What we are referring is to the fungal tissues. There are several modifications that you can see when in a different type of fungus, basically. The most common one modification that we see is a mycelial strand. The mycelial strands. The mycelial strands are nothing but aggregations of where the aggregate of a parallel or maybe an interwoven undifferentiated hyphae. So if you look into this hyphae, they form a parallel or they form an aggregation along with it where you cannot differentiate them. I mean, you cannot differentiate from one hyphae to another hyphae. That is basically called as a mycelial strand. That is, they are interwoven or they can be in parallel or they can be as interwoven undifferentiated hyphae. Okay. Another modification that is seen in case of the fungal tissue is the rhizomaph. The rhizomaph. Rhizo refers to root, morph refers to the shape, root shaped structure, basically. We can think of like that. This is a type of fruiting body where you can see that there are the mycelia is interwoven. It is a long root or a rope-like structure. Okay, you can see that there are the mycelia grows like a rope or a root-like structure, and it is basically a resting structure. Basically, what you can understand is it is a resting structure made up of a packed parallel or an interwoven, a parallel or interwoven parenchymatous tissues. A parenchymatous tissue or the parenchymatous mass of hyphae, and these normally often have a thick wall or a thick hard cortex. Thick hard cortex. That is why it is called as a resting structure. So what is a rope-like structure which is made up of a parallel or an interwoven parenchymatous mass or parenchymatous mass of hyphae with a hard cortex. This is called as a rhizomaph. Rhizomaph is nothing but a modification of your fungal tissue. Okay. And another one we have is the rhizoid. Rhizoid is again a short root-like filamentous outgrowth of the thallus. Basically, if you see, if you have a spongiophore, you will have a root-like structure, a short root-like filamentous outgrowth of the thallus, where the rhizoids are basically helping in anchoring to the surface or to an attachment to an organ. And also, basically, they help in absorption because they are in contact with the host. So, where they can help in absorption of the nutrition. So, there are two major roles that we can understand about the rhizoid. One is they are root-like filamentous outgrowth. Number two is the rhizoids serve as an anchoring agent or attachment to organs, that is to the surface, and they also help absorption of the nutrients because since they are attached to the host surface. Okay. And there is another type of fungal modifications that can be seen as a fungal tissue fungal tissues are also called as a plectenchyma, where the higher fungi, they organize into a loosely or a compact woven tissues. Loosely or a compactly woven tissues, which is called as a fungal tissue, or it is called as a plectenchyma. There is two different types when we talk about the fungal tissues. That is, one is called as a prosenchyma. A prosenchyma. A prosenchyma is basically, I can call this as an individual tissues, which are basically tissues in which an individual hyphae, they are interwoven. Individual hyphae, they lie parallel one to another, with the cells elongated, and they are distinguishable. Basically, if you see, they are distinguishable. Whereas in the other case which we were talking, you are unable to differentiate between one hyphae to another hyphae. But here you can, in case of prosenchyma, can differentiate between one hyphae to the another hyphae. Most often they lie parallel. They lie parallel to one another, with the cells elongated, and are distinguishable. The second type of fungal tissue, or the plectenchyma, is a pseudoparenchyma, or it is also called as the false tissue. It is also called as a false tissue. So these are basically a closed, compact, more or less they are isodiametric in shape. More or less isodiametric, and they resemble similar to that of the parenchyma cells of the plants. They resemble the parenchyma cells of a vascular plants. So the pseudoparenchyma is a close packed, more or less isodiametric or oval shaped, resembling the parenchyma cells of your vascular tissues or the vascular plants. So these are the common modifications that we see with respect to the fungi. That is, we come across a rhizomaph, we come across a mycelial strand, we come across a rhizoid, we come across a fungal tissue or a plectenchyma. Under this, we have a prosenchyma, which is loosely arranged, which lie parallel, where the hyphae lie parallel to one another with the elongated cells, and they are distinguishable. The pseudoparenchyma, which is false tissue, which is closely packed, where you can see that these are closely packed, which is oval or isodiametric, and they resemble to the parenchyma cells of a vascular tissues. Other type of modifications that we often see in case of the fungi is anastomosis. Anastomosis is basically what we call it as a fusion between two hyphae. There is a fusion between the branches of same or different hyphae. In exchange of the nuclei. So anastomosis is the fusion between the branch. So okay, two hyphae. In case of Rhizoptonia solani, which normally forms the anastomosis. There is another modification that is called as a clamp connection. A clamp connection. A clamp connection is basically a kind of, it is just like a buckle-like structure or a bridge. A bridge or a buckle, hyphal protrusion. Okay, you can see this. This is a hyphal protrusion, which is like a bridge connecting from one mycelia, one cell to another cell. You can see that from cell to cell contact. So basically, this is a hyphal protrusion or a bridge or a buckle-like structure, and formed by the fungus, mainly the Basidiomycetes. That is your mushroom fungi at the cell division. And this helps in connecting to the new divided cells. The clamp connection helps to connect the newly dividing cells, because to again exchange the nuclei. Okay. And I was mentioning most oftenly as something called as a conidia. Conidia are just like a spore, or I can call this as a seed. What is the difference between a seed and a spore? So both are having capability to germinate. Only thing that we see is in case of seed, you have an embryo, but in case of a spore, it is completely the protoplast. No embryo, completely it is a protoplast. And both germinates provided with a moisture and congenial condition. Normally, if you see, the conidias are of 2 to 150 micrometer in diameter. Okay, the conidias, which are, we can call this as a spore, or we can call this as a conidia. Sometimes we can call this as a sporangiospore, conidias, and so on. These conidias are different in shape and size. As I told, it varies from 2 to 1 micrometer in diameter. They are sometimes spherical, most often, or oval in shape, or sometimes ovate in shape. Pyriform type of are being observed, or orb pyriform. These are the different shapes I'm referring to. Sometimes ellipsoid shapes. Ellipsoid. Some of them are cylindrical. Some of them are like a filiform. You can see like just like a filiform. A filiform shape. Most often they are found without the appendages. Some of them also has an appendages like this. Some of them do have a kind of appendages. Most of them they don't. They can be a uni or a multicellular. Uni or a multicellular. Often they can be found in as a single, or they can be formed in chains. So this is the way how you can define a conidia or a spore. Okay. Conidia is a propagating structure, basically, wearing the shade, with respect to the shade, with a wide range, having different shapes, and having a uni or a multicellular, sometimes in single, or they're forming in chains. So this conidias, they originate. Where does they originate? They have to originate in a fungal structure. Either it can be on the mycelium or in the fruiting body. Okay, based on the origin of so-called as the conidia, where does they originate? They can be called as different type of conidias based on the origin. Again, I repeat. One of them is called as a blastospore. The blastospore. Conidia which are born as buds. Okay, they are born as buds, and once if they are mature, they cut off or they are blown off, and now they are called as a conidia. This is called as a blastospore. That is the spore born as a bud and blown away. That is called as a blastospore. And the another type which originates as a gangliospore. As a gangliospore. So here the conidia from the swollen tips. That is, if you see some cases, the swollen tips of the conidiophore. So this is the conidiophore, and this is the swollen structure of the conidiophore or an hyphae. So this will cut off and behave like so-called as a gangliospore. And also we have another one that is called as a porospore. The porospore, where the thick-walled conidia. So there will be a thick-walled conidia which form through so-called as the small pores that exist in a conidiophore. Through one or more small pores in a sporophore wall, or this sporangiophore or a conidiophore. Through the pore itself, they form a kind of the spore, which is a thick-walled structure. This is commonly seen in case of the alternaria or even the corollaria. So these are known to form the alternaria and the corollaria. They are known to form the porospores. The fourth one, which is called as Phyllospore, the Phyllospore. This is a very important type of spore, very commonly produced in case of the trichoderma, which is a very good biocontrol agent. Okay? So here the conidia are born on a structure called as a phyllids. So if you look into the hyphae, there will be a kind of a structure on the hyphae, which is this is called as a phyllid. The phyllid, which is a short unicellular oval or a subcylindrical, or I can say it is as a flask-shaped structure with the base swollen. The base will be oftenly swollen, and a narrow distal neck, a narrow distal neck, and on which the spores are produced. On which the spores are produced, these are called as a Phyllospores. The Phyllospore is the one with the conidia borne on a phyallid. And the phyallids are a short unicellular oval, often swollen at the base and distal neck, and on which the spores are produced. Those are called as a Phyllospores. And the example is the Aspergillus, or as I said, the Trichoderma, which is a very common biocontrol agent against many soil-borne diseases. Orthospores. The orthospores. Here, ortho refers to joints, where the conidia without a conidiogenous cells, that is, your hyphae, when it is cut off, so that is the one which is called as a orthospore. So the conidia without a conidiogenous cells, that is, without the conidiogenesis, or forming like a conidia or a spore, such a type of spores are called as a orthospores. And the other one is a Rhizomaph, where the conidia borne on a little pegs. If you have a kind of hyphae, there are small pegs which are formed, and on which the conidia are formed. This is basically, the small peg is called as a stigmata. The stigmata on a conidiophore, where you have a short peg-like structure that is called as a stigmata, on which the spores are produced. That is, that type of spore is called as a Rhizomaph. And this type of Rhizomaph is seen in case of botrytis. Is called as a botrytis. Genus is known to produce the Rhizomaph. And lastly, the Spores. The Spores. So here the Spore, the conidia are borne on a blown out ends of the hyphal tips. Let, for example, if you imagine that there is a hyphal tip, and this hyphal tip is blown off, cut off, and from there you will see that there is the spores that are coming out. Bilateral protrusions and cut off by a septum. So this type is seen in case of Trichothecium, the genus. That is, the conidia borne on the blown out ends of the hyphal tips, where they grow as a protrusions, later cut off by a septum. This type of spores are called as a Spores. And based on the liberations of the conidia. Liberation is how they eject out, or how they come out as a conidia. So based on the liberation of a conidia, there are several types of conidias that we can think of. There are two major types that we can see. One is schizolytic type. The second one is the rhizolytic dehiscence. Schizolytic dehiscence and a two halves of the double septum, they split apart and break the middle lamella. Okay, two halves of the double septum split apart by breakdown of a middle lamella. This type of dehiscence is called as a schizolytic dehiscence. That is, they become two halves of the double septum, split at a middle lamella, and they will be blown out or broken down as at the middle lamella. In case of rhizolytic, again, if you see, where the outer wall of the cell beneath the conidia. So here you can see the outer wall beneath the conidia, between the conidia 4, it breaks here. So it breaks and this becomes an independent conidia. That is, the outer wall of the cell beneath or between the conidia, they break and they behave as a independent conidias. This type is called as a rhizolytic dehiscence. Rhizolytic dehiscence. And conidiophores. What are conidiophores? See, there are two different types: the conidiophore and a conidiospore. Okay? If you have to ask me, this is the conidia or conidiospore, S-P-O-R-E, whereas this is a conidiopore, P-H-O-R-E. So there is a lot of, when I'm spelling out, please make corrections. What is a conidiophore? The conidiophore is, it can be a short or a long, or it could be branched, which could be a simple, or it could be a septate or an aseptate. And very important is, it is distinguishable from the hyphae, because your hyphae also appears same, but it is distinguishable where you can distinguish from the hyphae. That is called as a conidiophore. Conidiophore is a short or a long. It can be branched or it could be a simple. It could be a septate or an aseptate, but it is distinguishable from the normal hyphae. Okay. The conidiophores can be of different types. One is called as a holoblastic. The holoblastic conidiophores, where inner and outer membrane of the conidiophores, okay, the inner and outer membrane of the conidiophores contribute for the conidium formation. That is, inner and outer membrane of the conidiophore contribute for the conidium formation. Such a type of one is called as a holoblastic. That is, whole entire thing is responsible for the formation of a conidium. Another one is a enteroblastic. So in case of the enteroblastic, only the inner portion of the conidiophore or the conidiophore helps in formation of a conidia. Only the inner portion, the inner wall, helps in the formation of a conidia. And this type is seen in case of Helminthosporium and also the Aspergillus. That is, the inner wall helps in formation of a spore or conidia. The haloblastic example would be the Cladosporium. The Cladosporium is the examples that we can see. Okay. So this, when we talk a little bit more about asexual reproduction, how these are different. Whenever we talk about asexual spores, you will come across either a conidia or a zoospore or an aplanospore. A simple or a branched spore bearing hyphae is often called as either a conidiophore or the sporangiophore. Okay, this is basically a seed. The sporangia is a seed. Porous or pore is a bearer or a holder, I can say. Some are branched, some are filamentous, I can say. So basically, if you understand the spore as such, as I was talking a little bit earlier, which are a propagative. First thing is they are propagative. They are propagative in nature. They are reproductive in nature because they have a capability for them to reproduce. They are reproductive. And also they are survival units, that is able to survive for a longer time even in absence of the host. Survival unit. And this can separate, or they can separate out from the hyphae or a sporangioid cell, and they can grow independently. You can see that they can grow, they can produce, grow, germinate and produce the hyphae. And the spores can be of endogenous or they can be exogenous. So both types, they can be endogenous type or it can be an exogenous type. What is this endogenous and exogenous refers to? Endogenous spores, basically, they are formed within a swollen-like structures or a sac-like, swollen sac-like structures. And this swollen sac-like structure is called as a sporangium. Okay. Angium refers to vessel. Angium refers to the vessel, which can be the terminal portion or it could be at the intercalary, which could be at the terminal. You can see here the terminal, or it could be intercalary. Sometimes, please make a note that this entire portion can be called as Gautami. Can you mute? Sometimes this entire sporangium can behave like a spore. That is called as a sporangiospore. That is, entire sporangium can behave like an independent spore, that is called as a sporangiospore. Or aplanospores. Or so if it is a motile, again make a note. Again, if it is a motile, it is a zoospore. If it is a non-motile, it is called as a aplanospores. Zoo refers to animals. Talking about the exogenous spores. Exogenous spores. All asexual spores except sporangiospores are called as conidia. All asexual spores except your the sporangiospores, they are called as, commonly called as conidia. As I mentioned earlier, conidia is varying in shape. They are formed as branched or unbranched hyphal tips, and which are borne on a conidiophore. In some fungi, conidia are formed by the transformation of the existing cells. Sometimes what happens is the conidia can, for example, if you have a particular and behave like a conidias. And when the parent hyphae decay, when the parent hyphae, when this particular portion gets decayed, and now this can be called as the Thallospore. That is, the transformation of the existing cells of a thallus, and when they are set free, and when the parent hyphae degrades or decays, this type of spore is called as a thallospore. There are two different types of thallospores that have been seen, which I already mentioned about it. There are two types of kinds of thallospores. One is the arthrospore. One is the arthrospore, or it can also be called as a oidea. Oidea. Arthro, as I mentioned, it is a joint, are produced by the fragmentation. Already mentioned that whenever there is a fragmentation of a particular hyphae, or the apex of the hyphae, or the tip of the hyphae, and when they are forming a round shaped structure, separates out, that is called as an arthrospore. The second type of the thallospore is Climato Spores, which I already mentioned about it again. I would like to just again say that one of the cells of the hyphae, the cell wall becomes thickened, either terminal or intercalary, or the cells become thick, whether it could be a intercalary cells or it could be a terminal cells. Often they can be seen in the form of a chains, and this helps in resting. This is a resting asexual spore. The Clematospore is a resting spore produced to survive under the adverse conditions. So this is what I can say as different type of modifications and the types of spores that are produced by the fungi. So there are spores, types of asexual spores based on the shape. Types of asexual spores based on the shape. Number one, which is called as the amirospore or amirospore, where this is the conidia, which is a continuous, what I can say, it is basically a spherical or sometimes an ovoid type, or it is basically a cylindrical type, short cylindrical, and often it is a continuous one, conidia. All these are conidias, but are of, you can see, of different shapes. Each species are known to produce different shapes and sizes. Second type is the Allentospore. Allentospore, basically, which is the shape, which is an allentoid shape, where I can say that they are cylindrical. They are cylindrical, or sometimes they are slightly curved. Allentoid is slightly curved, and these are basically having hyaline to pale. Hyaline is colorless, pale, so the color is hyaline to pale. In case if it is an a colorless, they are called as Hyospore. And if it is a colored one, they are called as Phaeospore. If it is a color, if it is a hyaline, it is called as a Hyalospore. And if it is a pale color or a colored one, it is called as a Phalospore. And the third type is the Didymospore. The Didymospore, where Didymo refers to the septation or two Didy, which are ovoid, ovoid in shape, slightly curved, elongated, you can see, but they are two different septations can be seen. You have a septation, one septa is commonly seen. Hyalodydyme means it is a hyaline. This type of is a colorless. And Phaeodidyme, which is a colored. The fourth type is Fragmo Spore. The Fragmo Spore, where the conidias are basically oblong. Having septations or a transverse septations, I can say. A transverse septations. They are transversely septated, oblong, two or many septas, transversely septated. Hyalofragmospore or hyalofragme means hyaline. Phaeofragme